Okay, we're going to begin a section on nervous system. And to begin with, we'll go back, way back, and then pick up where we started forming neural tissues. Remember, this was amniotic cavity and yolk sac space down below, and we said that our entire embryo body would develop from a segment right in here, and this plate between the amniotic cavity and the yolk sac right across here, we call the embryonic disc. The embryonic disc. Outside of our ectoderm of our amnion, we had mesoderm, and then we had a chorionic membrane there, chorionic villi in the back. So anyway, we want to see what's happening right here in the origins of the nervous system. Looking down on that embryonic disc from above, this first picture represents the early formation of a neural plate. Neural plate ectoderm, that little line I drew in there represents that primitive streak. And you recall that the infolding of cells along that primitive streak forms the mesoderm that spreads out underneath there. So we had spreading mesoderm going out in different directions around between the ectoderm and endoderm layers. And if we were to cut through this here, we would see that in the middle there was a thickening of the ectoderm, making that neural plate. So first it's just a flat layer of ectoderm on the surface. The mesoderm spreading out then underneath of that, and then our third layer would be the endoderm tissues that form the gut. In our next picture here, our neural plate has advanced a little bit. We no longer think of it as a neural plate. It's forming what we call a neural groove. There's a depression down the middle, still have that thickening in there. But in profile now, that early neural tissue forms this neural groove. Sometimes we refer to these raised areas on either side as neural folds. To the side because they will eventually fold up. So that's the next stage. We had the primitive streak in the back. It eventually kind of closes up, and our neural plate is still developing back in the back. So things begin to develop at the front end first, and then later on they develop further back. Now, two additional views of developments of the neural tissue in the middle of the embryonic disc. Here we see the beginning of closure of the neural tube, starting not all the way at the front, but a little bit behind where the brain is developing. So that neural fold, as it comes together, it's a little slower to come together in the front because of the enlargement happening up there, that brain development that's occurring up there. So it comes together first in this region, and then from that area, kind of back behind the brain, it kind of moves forward slowly in either direction. So here we have closure of the neural tube. In this section here, and here it's still opening, still opened. We still have our neural fold here, and the groove down the middle. And we see that's still open in the back as well. As it develops and the brain begins to form, we have the development of what we call neuromeres. We'll talk more about these in a minute. I'll just label them neuromeres for now. They are enlargements, three primary enlargements at first. Forming different segments of the brain. here we are a little later. We have a little more closure of the neural tube up into the brain. There still remains an opening to that neural tube in the front, and that opening in the front up here is called an anterior neuropore. And in the back, we have a posterior neuropore. Now another reminder of what happens in the development of the neural tube formation. Remember, it started out as a flat plate, and then it formed a groove, and then it comes together. These folds come together at the top, closing in that neural tube, making the neural tube structure. Eventually, the neural tube separates from the surface ectoderm here, and we end up with a tube that's completely separated from the surface. There are some tissues right in there where things come together, right in here, that become a separate set of tissues between the surface ectoderm and the neural tube, and we call those neural crest cells, neural crest tissues. So this is surface ectoderm. This is neural tube. Ectoderm, and this is neural crest. Ectoderm. Okay, so what we were just looking at, cutting cross-section to show the surface ectoderm, neural crest, and neural tube. We're now cutting along a longitudinal view instead of a cross-sectional view of that neural tube. So this is neural tube from front to back at an earlier and a later stage of neuromer development. Remember, those segments of developing brain were called neuromeres. So our first three neuromeres, when they first start to appear, are prosencephalon, then mesencephalon, and rhombencephalon. The front chamber up here is going to divide eventually into two. We'll look at over here, the telencephalon and diencephalon. The middle one, the mesencephalon, simply remains mesencephalon. And then the rhombencephalon back here divides into a metencephalon and myelencephalon segment. The space inside, this cavity that formed as part of that tube, is regarded as a neurocoel. We should have labeled that in the last picture where we had the cross-sectional view in there. But anyway, there's our neurocoel. Now, prosencephalon, as we said, divides into telencephalon. It becomes primarily your forebrain, your cerebral hemispheres, your two cerebral hemispheres in the front. Next is diencephalon. Diencephalon, that's your thalamic region, hypothalamus, thalamus, and epithalamus or pineal gland. Then is mesencephalon. It becomes part of a reflex center for both auditory and visual response. Now, divisions of the rhombencephalon include metencephalon, both the pons and the cerebellum of the brain are derived from metencephalon, and then finally myelencephalon. The medulla of the brain, or medulla oblongata, derived from the myelencephalon portion of this early five-part brain. Each of these, remember, is still referred to as a neuromere, so we have 5 neuromeres at this stage, derived from the early 3 neuromeres we see over here. Now we want to turn our attention to developments of the neuromeres, the neurocoel rather, the cavity inside the neuromeres. So we had our 5 neuromeres: telencephalon, diencephalon, mesencephalon, metencephalon, and myelencephalon here. But that cavity on the inside becomes our fluid-filled chambers of our brain. Cerebrospinal fluid is produced inside of our brain, fills these cavities up, but before they are the ventricles and passages we know in the adult brain, they are these spaces here. So from our developing cerebral hemisphere regions up in here, we get ventricles 1 and 2. Sometimes simply referred to as lateral ventricles because they are to either side. They eventually come together through 2 separate passages as these 2 chambers develop separately. They join together in the 3rd ventricle here. The 3rd ventricle. Between the 3rd ventricle and the 4th ventricle is a space that runs through the mesencephalon. That space that runs through the mesencephalon doesn't become one of our primary ventricles, but is known as the cerebral aqueduct, or aqueduct of Sylvius. We'll just call it cerebral aqueduct. So that runs between our 3rd ventricle and our 4th ventricle, which is right here. Beyond the 4th ventricle, which lies underneath the cerebellum there, beyond that 4th ventricle, this space down here simply funnels into the spinal cord as a space we call the central canal. Central canal. So these start out as neurocoel spaces, the cavities within the early brain, and as the brain develops, they remain as fluid-filled chambers, 1st and 2nd ventricles of the cerebral hemispheres, 3rd ventricle within the thalamic region of the brain, the cerebral aqueduct that runs through the midbrain, 4th ventricle that's underneath the cerebellum, and then the central canal that runs from the medulla on down through the spinal cord. Okay, I made a drawing of the brain here. It's not the greatest drawing, but it'll do for what we need to talk about. First of all, we know our cerebral hemispheres are a larger part of the brain, and I didn't draw them maybe quite big enough in comparison to the rest of the brain. But anyway, this part that's in this lighter color up here, this pink color, represents the part that's derived from the telencephalon, the most anterior parts of the brain. So anyway, all of our cerebral hemisphere we think of commonly as gray matter on the outside of our brain. Our thinking part of our brain up here, our voluntary functions are somewhere in here, our visual centers are somewhere back here. Auditory and speech centers, all those are in our cerebral hemispheres. The next part, the diencephalon here in purple, I've drawn in the little circle in the middle here. It's called the interthalamic adhesion, or simply the thalamic adhesion, sometimes called the massa intermedia. But anyway, it's this place where the 2 sides of the thalamus come together and meet in the middle. Surrounding that is an open space of cerebrospinal fluid. We'll talk more about that later. But our thalamic region is divided into a hypothalamus along here, the central portion of the thalamus we've already mentioned there, and then what's called an epithalamus. It really kind of tails off the back over here instead of sticking up off the top. But anyway, that's the pineal gland. Produces your melatonin for your sleep-wake cycle. So there's these two sections are derived from the prosencephalon, telencephalon and diencephalon sections. The next section would be the mesencephalon region, the midbrain section. There are two enlargements that stick off the dorsal side of the mesencephalon. Well, there are actually four, two upper ones and two lower ones. There's a left and right superior colliculus and a left and right inferior colliculus, those portions of the mesencephalon that are responsible for, like I said, those reflex responses to either visual or auditory stimuli. Behind the mesencephalon, we have the two parts of the original rhombencephalon, the first being the metencephalon. It consists of the pons on the ventral side and the cerebellum on the dorsal side. Cerebellum is fairly enlarged. It helps coordinate many of our normal muscular movements for things like walking and all kinds of fine movements, playing musical instruments, doing athletic sorts of things, many routine kinds of motions that we go through to eat or just write or whatever we're doing. They're coordinated through the cerebellum. The pons has a few centers that help with respiration. That's their primary contribution that we talk about. And then finally there's the medulla, the final portion of medulla oblongata derived from the myelencephalon of the brain. Many of your reflex functions having to do with coughing and vomiting and those kinds of things are centered in here. Some of your cardiac regulation centers located also in there. So several functions situated in the medulla oblongata. But remember that they were originally just 5 segments of a tube that formed the telencephalon, diencephalon, mesencephalon, metencephalon, and myelencephalon, portions of your original brain. Now, if we wanted to look at this and think about where our cerebrospinal fluid-filled cavities are located in here, it's a little bit complex. I'll try to draw them in brown here. If my brown pen works, let's see, yeah, this is going to work all right. So on both the left and the right side of the cerebrum, we have this complex kind of shape here where the two cerebral hemispheres have divided. There's one of these structures on the left and one on the right. So those are ventricles 1 and 2, or your lateral ventricles of your brain. They kind of have a tail that pulls around from the outside. It kind of swings out and then back toward the middle a bit. Next, there's a connection between these two and that space that I mentioned around the thalamus here, around that interthalamic adhesion. So in here we have the next ventricle, the third ventricle of the brain, right here. So both of the upper ventricles, the left and right ventricles, the lateral ventricles up in here, they produce some cerebrospinal fluid that drains down into the third. The third also adds to it some cerebrospinal fluid that will be moved into the cerebral aqueduct. And here's our cerebral aqueduct that runs through the midbrain portion, the mesencephalon. Then our fourth ventricle right here under our cerebellum, and then it tapers down into our central canal of the spinal cord down below there. So there's your fourth ventricle there. So here's lateral ventricle. Third ventricle. Cerebral aqueduct. fourth ventricle, and finally our central canal. There are 3 pores that allow fluid to move from the fourth ventricle space to the space that surrounds the brain and spinal cord. So we produce cerebrospinal fluid in our lateral ventricles, in our third ventricle, and in our fourth ventricle. We're continually producing more cerebrospinal fluid, and it exits that space through tiny pores that are below the fourth ventricle. There's a median aperture and 2 lateral apertures that allow that fluid to move out to the outside of the brain so that the brain and spinal cord are all encased in that fluid as well, surrounding the outside of the brain and spinal cord. That cerebrospinal fluid will be reabsorbed into the bloodstream with vessels that are found in the membranes that surround the brain and spinal cord. Now I'm going to attempt to show those same things on this model of the brain and another model of the brain here. But to begin with, all of this is cerebral hemisphere here. This white band across there connects the left and right cerebral hemispheres, so that's also part of telencephalon. All this is telencephalon material here. All the way back to here and up to here and around. So that's telencephalon. The diencephalon, this little brown dot in the area around it right there represents the thalamus of the brain. Hypothalamus would be below the thalamus, right through here, this tissue right along there. All of that is hypothalamus. That little bump right there is part of hypothalamus. The epithalamus or pineal gland is right there at the end of my pen that I'm pointing to right there. So anyway, that's all diencephalon. Diencephalon. And the third ventricle of the brain surrounds that space there. There's a cavity all around that. Third ventricle around the thalamus. Next, we have the mesencephalon, and in the mesencephalon, remember we had the 2 bumps. You have the bump there and the bump there, both left and right. Those are the superior and inferior colliculi that help coordinate those reflexes we talked about. And through that runs the cerebral aqueduct. That little groove running right through here is the cerebral aqueduct. So that's mesencephalon right in there. Beyond that, we have metencephalon, this region right here. It includes the pons, that little oval right here is the pons, and on this side we have the cerebellum. Pons and cerebellum, and between the two, underneath the cerebellum right here, we have this cavity, that's the fourth ventricle of the brain. Beyond that, this tissue down here, this is the medulla oblongata. Another model showing the same thing, just to the other side of the brain, just a mirror image there, but a different model. Again, we have the cerebellum, all telencephalon up here, which means cerebrum. Cerebrum, all the telencephalon up here, that connecting material in white. The lateral ventricles, 1 and 2, would be not in the cut plane right down the middle. You'd have to go off to deep into one side of the brain or the other to find the ventricles 1 and 2. But they would drain into the third ventricle, which would be right here. There's the thalamus, there's the pineal, and this would be the hypothalamus region right across here. So this is diencephalon in the region of the third ventricle. Next, this is the cerebral aqueduct. This is mesencephalon right in here. Our superior and inferior colliculi, that's what I'm trying to say, superior and inferior colliculi of our mesencephalon found right there. Next is the fourth ventricle under our cerebellum. Here's the pons region of the metencephalon, so that's all metencephalon region with the ventricle, the fourth ventricle, the cerebellum, and the pons. And then beyond that is myelencephalon, the medulla portion down here.